Okay, yeah, hello everyone. This week we are going to talk about the first bioinformatics problem, which is a really classical problem, uh, sequence alignment. So when we talk about sequence alignment, it's biological sequences, DNA sequence, protein sequence, and I'm going to first give you some examples. Why do we align sequences? Why is this biological requirement? What motivation behind it? For which tasks we use sequence alignment these days? And I'm also going to talk about one of the most popular sequence database search algorithms, like BLAST algorithm. But before that, we are just going to start with pairwise sequence alignment. And we'll see an algorithm which is an efficient algorithm that works in polynomial time based on dynamic programming. On the lecture slides on the course homepage, we have also an example code that aligns two given sequences from an input file like this. If you take a look at this one, these are two DNA sequences. Let me also. All right, so if we have these two, these are really two small DNA sequences. Most of the time, we are going to be trying to align DNA sequence of thousands of bases long. So this is a really simple toy example. And the goal will be, given these two sequences in the sequence alignment problem, this sequence may be from one organism or and it may be some part of a gene. All right. As I said, this is really a toy example. Usually, when we are aligning DNA sequences with each other, they are on the order of thousands of characters long. Each of these characters are called bases. Okay, so for example, kilobase is regularly the length of a gene on the order of kilobases. So each base here or nucleotide here, as you can see, is actually a nucleotide on the genome, on the chromosome, and it may be part of a gene definition. It may be part of some regulatory region. So these are different regions on the DNA, have different functions, different mechanisms. And what happens is that one of the reasons we want to align these sequences is that different organisms have similar DNA sequences. Certain genes, they are not unique to humans. For example, the hemoglobin gene. All the animals who have blood, the hemoglobin protein. So when we look at hemoglobin protein structure, its quaternary structure is a tetramer. So two alpha subunits, two beta subunits. We're going to talk about these when we talk about protein structures. But this protein is synthesized the definition for this, the recipe of how we're going to human hemoglobin is going to be made out, is going to be is in our DNAs. And there are these alpha subunits, beta subunits. There are DNA sequences that define. For example, let's take a look at. Let's go to UCSC genome browser and search for the hemoglobin alpha subunit, and let's see what kind of a gene structure it has. Genome browser, let's choose human, and let's choose a certain version of reference assembly. So this is, as if you look at this, these are just like the operating systems have different versions. These are the human operating system versions starting 2003. So the thing is that we are not evolve. It doesn't mean that humans are evolving. That's why it's changing. The reason we have these different reference sequences for human genome is that the sequencing technology gets more and more accurate. Right now, there is no single machine that can read the entire sequence of certain individual by just getting the chromosomes from a cell, reading it one by one accurately from beginning to end. There is no technology. The way the next generation sequencing technology that we use today chops up the DNA sequence into smaller pieces. It replicates them, and it gets lots of these small readings. So it's actually solving a puzzle. So there are algorithms that are used, really, string algorithms that are used to get these reference genomes, the entire sequence of the DNA of the human. So as these algorithms get better and better, certain parts of these puzzles are more difficult to solve. For example, if you're solving a jigsaw puzzle, if the sky is all blue, lots of repetitions, it will be difficult to find a puzzle piece to see where it fits, right? If it has more information, in other words, if a certain region of a jigsaw puzzle has a higher entropy, lots of changes there, if you get a puzzle piece that is going to fit such a location, it's easier to find. The same thing with human genome. There are certain regions of the genome with very low entropy, which we call low complexity regions. And when we get small sequences from those low complexity regions, it's really difficult to figure out where these short reads are going to fit. All right, so therefore these are like challenges to these algorithms. But as the algorithms get better, as the sequencing technology gets better, when these puzzle pieces, instead of being very short, if we can sequence longer sequences, it will also help us to figure out ambiguities. And with all these, as you can see, we have different versions of the human reference genome, and we don't have new versions in 2013, because this 2013 version, I think it's pretty accurate. Oh yeah, the Debrien graphs are used to find, so one of the ways to find these alignment sequence assembly is using a data structure, a graph data structure, which actually connects strings with certain overlaps, and having that overlap information on the edges is called Debrien graphs. You're right, I guess. So, and these are really time-consuming algorithms, and having novel sequence assembly these days is really difficult. So it means that if you don't actually see the picture, solving a jigsaw puzzle without your reference picture, just by having the puzzle pieces, is extremely more difficult compared to solving a jigsaw puzzle by looking at a reference picture, right? So these days, these reference sequences serve that purpose to find. We are sequencing individuals. The genomes are individuals that are humans, and these reference sequences serve as that picture, which is similar to the individual's genome sequence that we try to assemble. But since this December 2013 version, I think it's very accurate. There hasn't been some major changes that necessitated the release of a new version. So I was talking about hemoglobin. Hemoglobin. I think it's written like this, right? Alpha subunits. I think I misspelled it, or less hemo. The heme group. So yeah, hemoglobin. Globin. So human. Yeah, it's here. So it's hemoglobin. How long? Yeah, a good question again from Ömer is that how long does it take to sequence a genome today? It depends on whether it's a plant genome or an animal genome, or whether you are doing you're using a reference genome, like to sequence a human individual's genome. If you have the next generation sequencing technology already spit out those short reads. If you have the short reads already, you may actually, depending on the power of your machine, it may take an hour or so. But there are also cloud frameworks in which, there are supercomputers, clusters, which you can actually parallelize this job. One of those available platforms that I use a lot is the by Seven Bridges Genomics, the company Seven Bridges, the Cancer Genomics Cloud. So you can actually use certain tools on the cloud to make this faster. You can also run certain tools, these aligners, the assemble the sequence assembly with respect to a reference genome is also called alignment because it makes use of this alignment technology that I'm going to talk about these days. The sequence alignment is one of the major things that, because sequence alignment means that you have a puzzle piece. Your goal is to figure out where does this puzzle piece best fits on your reference genome. So you have one very short sequence and you have another sequence which is really long, 3 billion characters long, and the other is like 150 characters long. This 150 characters, where does it fit on this 3 billion character long sequence? Where does it fit best? So there will be differences. There won't be an exact match. But there are heuristics that are used here to make this process very fast. Because if we use dynamic programming to solve this, it's going to be much, much, much slower. Maybe it's going to take a month to assemble your reference, to assemble your sequence for this individual. But yeah, there are heuristics that are used here. There are certain indexing strategies. There is the Burrows-Wheeler transform. Let me write here, Burrows-Wheeler transform. I may also talk about this Burrows-Wheeler. And there's the tool that uses this Burrows-Wheeler transform is BWA, Burrows-Wheeler transform alignment. So BWA alignment, if you go and search Burrows-Wheeler aligner. Okay, so it gets these small short reads and try to align it against large sequence. Now, today we are going to see. So I was just going to try to show you, for example, this is the alpha subunit, subunit alpha 1, subunit alpha 2. Actually, there are on chromosome 16. There are two. Oh, there's also the gamma subunit. Yeah, alpha hemoglobin subunit. Another subunit theta. So it has actually homologs. I mean, there are which are called. So what we have here is we have similar copies of the same gene even on the same organism. But what we are going to do is, if I, for example, select this alpha subunit, we can also take a look at, so this is its gene structure. Let's zoom out. When we zoom out enough, so this is human hemoglobin alpha subunit 1. This is the genome sequence, certain parts of it. So these parts, they do not encode, they're called introns, and they're not used to they're not translated into protein sequences. So it's actually they're really also, it's just interesting. I didn't know this copy existed like this. So one of the copies is in the forward strand. All right. So it's in the gene is read from left to right like this by the polymerase, and when it's messenger RNA is built from this, it's read in this direction. But on the other hand, oh, it's also in this direction. So I thought this was probably, I think it was showing the start of exon 2. So this is also forward strand. These arrows showing that it's read from left to right. Let me zoom out a little bit more and move right. So yeah, it's right here. So it has 3 exons. If you hover on this, it's going to probably say exon 2 of 3. I don't know if you can see it. It's a really small pop-up. Maybe if you watch the recorded video later on YouTube on high resolution, you can see this is exon 2 of 3, and this is exon 3 of 3. So this is the sequence, and it's it goes from this. So this is like a scale, 10 kilobases, but it has certain similarities in different organisms. For example, if we look at mouse, dog, elephant, chicken, zebrafish, so there are the same. The reason that I was showing this was hemoglobin is a shared gene and it has similarities in different organisms. So which we call ortholog. If an organ, I mean, if there's a homolog means that they have the same function, they evolved from the same common ancestry sequence. All right. So in the evolution, but with certain speciation, certain events, evolutionary events, some of them are speciation, some of them are replication within the duplication within the same organism. But what happens was homology is a binary term. There is no, most of the time, this homology is used by biologists or other computer scientists as well as to indicate similarity, which is of a certain degree. Similarity has a degree. You can say these two sequences may be 70% similar, 80% similar, 40% similar, but homology is something that we infer, and we may not always, especially for proteins which share little or genes that share little sequence similarity. Homology sometimes is something we guess, and we may not be 100% sure. But it's either there, they're either homologs or not, all right? Which means that they either have evolved from a common ancestor, which means that they may serve for similar functions or not. So that's why sequence alignment can be used as an indicator of homology. Higher percent sequence similarity may indicate these two genes are homolog or not, but really what I want to say is that different organisms have the same genomic sequences. They share them, and there are high similarities between certain genes. We share similarities to single-cellular organisms even in certain genes. So sequence alignment, its main purpose is given such one sequence from one organism and given another sequence from another organism which may belong, finding which parts have been retained through evolution, which means that alignment means that the process of aligning is to map, to produce a one-to-one mapping between the nucleotides here and the nucleotides here. And we also do alignment not only on the DNA sequence, but we also alignment on the protein sequence where we have these 20 amino acids. And again, the purpose would be the same thing. You're going to match amino acids or nucleotides here on the first sequence to the other one. And this mapping have to be one-to-one. And since they are of different lengths, some of them, so the evolutionary events that we will talk about, may be insertion of, let me zoom in here too. So insertion of a nucleotide at that spot in the second sequence. Or you may also think this as maybe this nucleotide is deleted on the first sequence. So there may be insertion deletion events, which are sometimes called indel events together, because they are looking at from different perspective, which sequence perspective you're looking from. It may be an insertion from the sequence of from the perspective of the second sequence, but from the perspective of the first sequence, it may be deletion. Okay, and what we may have actually here is that can I say, for example, since this one is one nucleotide longer, this one is one nucleotide shorter, there is only one indel event. Is this statement correct? Can you write yes or no? No. Thank you. So yeah, it was a trick question because what we may have here is that I can put here lots of, for example, I can put something here like this and something here like that, and insertion and deletion here, maybe another one. So I can have an alignment that is going to match the sequences underneath here like this. Maybe this whole chunk may be inserted here. It may be missing in the other one. And this whole chunk here may be inserted by evolution. So here, During the alignment, we may insert and delete certain parts of the sequences to different places. All right. So, but at the end, our goal is to try to figure out the sequence alignment problem's main purposes, to figure out what may be the biologically most viable, possible way that these two sequences are related. Okay, so this is a really important statement. So the purpose of sequence alignment, the optimization goal, come actually from the biologist's point of view. It's not like the edit distance, where we, after we define this computational problem, saying that what is the minimum number of insertion deletion operations so that the number of matching characters that are underneath, that are mapped to each other. So but if we map two nucleotides with each other, okay, for example, if this T is mapped with this G, what will happen is that it will be a mismatch, right? So, but if this C is matched with this C, it's a match. So two characters that are associated with each other, that are mapped to each other, it may be a perfect match, it may be a match, or it may be a mismatch. So we may also pose this problem, maximize the number of matches and try to insert insertion deletion events such that these maximum match the matches in these mappings are maximized. But this will make it a really mathematical computational problem. But from the biologist's point of view, it may still not be the most accurate, especially in the case of protein sequences. Again, let me repeat the statement. From a biological point of view, our goal is, given these two possible homolog sequences that share some common ancestry, we try to understand what evolutionary events have made these two sequences different from each other. All right, where did we have an insertion in evolution? Which sequences are deleted in the other one? Which part of the sequences are mutated to something else? There are mutation events, which means that those mapped elements is going to be a mismatch. There may be a mutation. There may be insertion deletions. Black blank board. It should be this notepad you should be seeing actually. Yeah, it should be normal. So you should be, if you're getting my shared screen correct, you should be seeing this notepad input. It's black mostly. I can actually change the background to something else, but you should be seeing this notepad. I'm going to go over the slides in a couple of minutes, and it will be you will also see a nice white background. But yeah, the main thing is we need to really biologically, our goal is to figure out what is the biologically most possible way these two sequences are related. For that, especially in the protein sequences, we are going to see that certain mismatches are more likely to be observed in nature compared to other mismatches. For example, it may be from a computer science point of view, two strings. Let me show you some protein sequences here. Yeah, look at these. So these are protein sequences. It's no longer ACGT, as you can see. We have 20 letters here. For example, this K and W, it's a mismatch, but it may be a less likely mismatch than this H and D. All right, so the mismatches that we are going to have, the mutations, are going to have a different possibility of being observed in nature. So if you're looking for a biologically most possible way to relate these sequences, how this happened, these evolutionary events happen, we need to actually take into account not just mismatches and mismatches. We need to somehow put into our algorithm these biological domain knowledge, and they are going to come in the case of scoring matrices. We are going to talk about those. And in 1970s and 80s, different research groups worked a lot on how to represent that information, how to build those those scoring matrices that show these different relationships, different matching possibilities, mismatching possibilities, and they gave them some integer scores so that they can be directly incorporated by an algorithm that is just counting, accumulating the match scores or mismatch scores. So we are going to talk about these, okay? But just I wanted to this is really important because I just wanted to emphasize what is our goal here in sequence alignment. In sequence alignment, we are given two sequences, and our goal is to figure out what is the biologically most possible way that these two sequences are related. In that case, we can see some evolutionary events, certain insertions and deletions. But another thing is that when we are aligning, for example, hemoglobin sequences of 20 organisms on top of each other, which is called a multiple sequence alignment. Sometimes we're not just doing two sequences, pairwise alignment, but we're aligning multiple sequences on top of each other. And this is actually the algorithms that we develop in pairwise sequence alignment. Their complexity is exponential on the number of sequences that are aligned, which means that if you're aligning a lot of sequences on top of each other, it's not going to be a viable, or it's going to be intractable. It's with this exponential algorithm. But what I want to say is that when you multiply align all these sequences on top of each other, you're going to see that certain regions are conserved. Conserved means that it doesn't change. They remain same among all those 20 different organisms. Okay, certain regions, you will see no mutations, no insertions, no deletions. There will be blocks of conserved regions that are the same in all 20 sequences, or that are very similar to each other. Now, we may ask this, at this point, we may ask ourselves, why is it so? Why do those regions are the same, shared among those 20 sequences? Can it be they are biologically, functionally more important? Because in evolution, a change here, a change in those places, a mutation in those places cannot be tolerated at all. The organism maybe dies if there's a mutation in that spot. So, therefore, throughout the evolution, those places remain as similar in all these different species, and therefore the organisms continue to live. So it may, such conserved regions hint, they may give, they may point to us these conserved functionally important regions, and they are actually used. Yeah, this is like positive selection, exactly. Which, I mean, if there's something bad happens at that point in evolution or phylogenetic, this is also called positive selection. Certain parts that are same, that are functionally important, they are observed throughout different lineages. So it's really, sequence alignment help us find these regions. Sequence alignment help us to align an individual's genome against a reference genome. With sequence alignment, we can also infer homologies. For example, if you have a new organism that is just newly sequenced, how can you figure out which genes? First, you identify the genes, and the next step is, what are those genes' functions? What are those genes? Is there, for you get the gene sequence and search it against the database by using tools like BLAST, and if you get a hit, and if there's a sequence similarity of 60%, 50%, it may be a strong indication that the gene you have found in the database is an homologue of this new of the gene that you found in your newly sequenced organism. So you can assign, then you can transfer function, these known functions that you know from organisms in the database to this new organism. So it's used for function prediction a lot. It's used for so 50% sequence similarity allows you to infer homology, basically. But sometimes it's not sufficient. We know that there are homologue genes, which means that there are genes in different organisms, ortholog genes that are genes in different organisms, which have only 5 to 10% sequence similarity. So if you look at their protein sequences, it's diverged a lot. So it's different. It's really different. It's similar only 5 to 10%, but they may still have the same function. Okay, so this is not an if and only if relationship. One way of the if is correct. If two sequences share like 60-70%, if they share a lot of sequence similarity, they most likely share similar function. Again, this is all the things in biology. When I'm making these statements, there are always exceptions. Okay, so you may have 60-70% homology, but those different parts may also make the actual function diverge a lot at different functions. So there may be exceptions. But just all the statements that I'm saying here, take them with a grain of salt and just think of them as some generalizations that work 90% of the time, 80% of the time. So again, let's come to the first part of the if. If we have lots of sequence similarity, they are probably evolutionary related, which means that they probably have the same function. So but the other side of the if they have biologically the same function, does it mean that they should have 60 or 70% similarity? No. That other arrow is less likely to occur, so it's not an if and only if type of thing. So therefore, there may be functionally similar sequences. This does not imply that they need to have high sequence similarity, because there are lots of examples. This is not like an exception. There are examples in which sequences share. We know that they are functionally similar proteins, but they share very little sequence similarity, especially in certain globin family proteins. There are a lot of differences. I'm going to talk about these later. In those sequences may not be shared, but if we look at their protein structures, we see that protein structure is more similar. Although the sequences are different, the resulting protein structures may be similar, more than a certain threshold, which may make them functionally related. Okay, but yeah, this was just basically, the first part was just basically the motivation, why we need to align sequences, because DNA sequences, genes in different organisms, and the resulting protein sequences, they share similarities. And our goal is to figure out what the most possible way to just propose, whoever is analyzing these sequences, the algorithm is actually, the result again is not 100% certain, the result of these algorithms, but they try to output the most possible way to relate these sequences. And this relation, as I said, is done by mapping, a one-to-one mapping between a nucleotide here to a nucleotide here. That one-to-one mapping sometimes may be a mismatch in the case of this TNG, and sometimes may be a match in the case of this, and sometimes we may have insertion events and deletion events. So, and if you then see what is the search space, if you look at all possible, what is the number of all possible alignments? I'm going to talk about that in a couple of minutes, but you're going to see that the exhaustive search of all possible alignments and trying to figure out the one which gives the best score. So we need to have some kind of a scoring mechanism to compare an alignment like this. For example, is this a better alignment? If one program outputs this one and the other program outputs another one, which is another program may output something like this, okay. So which one is the better alignment? So we need to be able to have a scoring mechanism to compare, to score these alignments. Again, the biological potential, possibility should be somehow modeled with a mathematical computational scoring mechanism. So this is by itself a problem. What kind of a scoring mechanism would best capture what the biological relate possibility? So that biological possibility is difficult to measure as a number quantitatively. So we usually have, as computer scientists or as biologists that work in this area that build those scoring matrices, their goal is to again find heuristics to define certain, make abstractions, assumptions. We make lots of assumptions. We make certain abstractions to compute a score to see how potential is this one and how possible is this one. So that's basically the score of an alignment. And if you have a way to compute the score of an alignment, by the way, where do these pluses came from? Oh, so it should be, yeah. The score of an alignment, if you have a way to compute those, one algorithm could be try all possible alignments, compute the score, and just as the algorithm, output the one with the maximum score. Now, that if we have that, we are going to start something like that. Unfortunately, I can just directly tell you that the number of possible alignments is exponential, 2 to the n. It's proportional to the 2 to the n when both of the sequences are of length n. All right, so even with sequences of just 50 nucleotides long, 2 to the 50 is a very, very, very large number. So I usually give this example. I don't know, I forgot where I heard this from earlier, but the number of atoms in the universe is like on the order of, that's the predicted number of atoms in the universe is like 10 to the 80. Okay, 10 to the 80. Let me write in the chat box, 10 to the 80. So this is the number of predicted number of atoms in the universe. So it's roughly 2 to the 2 to the 150, or so 2 to the 3, yeah, 2 to the, you should multiply 8 with 3, like it's like 2 to the 200, something like that. Yeah, exactly, 2 to the power 260. So it means that if you try to align two sequences of length 250 bases each, as I said, we are trying to align thousands of long sequences of length, thousands of amino acids or nucleotides, going over all the possible alignments is really not possible. Now, how can we figure out a way, how many different ways to align these two sequences? Now, I'm going to just quickly try to show you why it is exponential. Now, I'm going to show you a different way to show how these two sequences that I have here are aligned. By writing one of the sequences, actually, let's see, one of the sequences I can, so AGGCTA. I'm just going to write AGGCTA by writing one sequence here, and the other sequence, TAGCTA. TAGCTA. Now I'm going to define, so this is like a grid, like a matrix of cells, all right? Now I'm going to, this model is going to serve a nice purpose for us. We'll be able to do lots of analysis on this one. But now I'm going to define some path from the top left cell, okay? Actually, if we have it like this, it's if we just put the first characters here as the first row and first column, the top left cell here actually restricts us to align the first character against the first character in the other one. So this is what I mean. Visiting this cell means that you're aligning these two things. So what I'm going to do is I'm going to put an additional row, an additional column here, like this, okay, which do not correspond to the first sequences. And I'm going to start actually from this corner. And I'm going to define a path from this top leftmost cell to the bottom right cell, okay? And the path is going to be something like this. We are allowed to go diagonal So these are the three. From a cell, you're only allowed to make one of these three moves. Okay, we'll be able to move with these three moves only. Now, an alignment, any alignment actually, corresponds to a path from the top left cell to the bottom right cell. For example, if you just now here's an interesting question. If two sequences are of different length, is it possible to move just with the diagonals and reach from the top left to the bottom right? If they're of different length, it's not possible, right? Only in a square matrix like this. Square matrix means that if the length of these two sequences are equal, you can just move diagonal, 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 and come from the top left to the bottom right. If they are of different length, you have to make one of these moves, either move right or move down, all right, to get from top left to bottom right. Now, for example, an insertion in this sequence will move to a successive movement to right here. What does it mean? When you're moving, when you're coming to this cell here and you're moving right, it means that you're consuming amino acids here while you're moving to the right on the first sequence, but you're still at this sequence T, which was actually consumed at the previous step when you move diagonal like this, which means that you're putting those dash symbols on the second sequence. So a movement to the right is going to correspond to an insertion to the second sequence, or a deletion from the first sequence, again, depending on where you're looking for what your perspective is. And moving down like this is going to correspond to putting some dash symbols, so inserting something here in this sequence and deleting certain parts is deleted here. So by just any alignment, as I said, can be represented as a path from the top left to the rightmost. Now, the question that I ask, how many different alignments, possible alignments, exist between these two sequences? For example, one alignment could be, look at this interesting, trivial, but really not useful alignments. You can move right, and you can move down, like this. So what kind of an alignment is this? It's like, it will be an alignment that is like this. Let me get back to these ones. It will be an alignment. You can immediately guess actually what kind of an alignment it's going to be. It's going to be something like this. You put gaps in the first one, you put all gaps in the other one. Yeah. Look at this alignment. This is also an alignment, right? I aligned these two sequences. After the alignment, the aligned part should have the same length of sequences, but the total length of the alignment means that it's the same on the top and bottom sequences, but this is also a valid alignment. Or I could just switch these insertions and deletions, vice versa. I could first do the other one and do this one next, and then it will then correspond to an alignment that's moving down here first and then moving right all the way here. But in any one of these alignments, when we try to align the entire sequences with each other, this is called global alignment. Okay, so this term, global alignment, refers to the fact that in this alignment, we cannot just ignore parts of the sequences. The entire sequence, the first sequence, we try to align it to the entire second sequence, which means that we are trying to start from this top left cell and move to top right cell. Now, try to write a program to solve this problem. If I give you, actually, I gave this when I was teaching algorithms class and when I was talking dynamic programming. This was the simple examples that I gave earlier. How do you figure out how many different possible ways there are? What is the number of different paths you can take from top left to the top right? Does this seem like a small number? No. How do you compute this? Here's how we can compute it actually. I'll directly tell you the solution. If the number of possible paths that end here, let's call it P, Okay, let's call it Pij. Pij, let's define this number Pij as the possible number of paths that starts at the beginning and that end here. At the end, our goal is to find P and M, where N is the number of nucleotides in the first sequence and M is the number of nucleotides in the second sequence. Okay, so our goal is to figure out what P and M is. And what Pij in this case, if you define Pij to be the number of paths that start at the top left and end at here, Pij is simply, can you tell me what Pij is in terms of this one, and this one? Okay, if somebody tells you the number of paths that end here, because if you come to Pij, the paths that come to Pij has only 3 possible previous steps, right? I mean, is it possible to come to Pij directly from jumping from this cell to here? No. If you come to Pij, you should either follow this path, or this path. Now, if, and which in this case, these are Pi minus 1, J minus 1, and this is Pi minus 1 Pj, and this is this is Pi Pj minus 1. So in other words, Pij, and what is the total, if somebody gives you the total number of paths that end here, and end here, what is Pij? It's simply the summation of all those. Pij minus 1 plus Pij minus 1 plus Pi minus 1 J. All right, so this is the recurrence relation that we can use in our dynamic programming solution to solve what P and M is. If you write, okay, so this is, as you can see, this is a recursive function. If you try to write a function that says count, and in which case 2 arguments, n and m, and if you call this count nm function, and write a recursive function saying that count nm, I mean, the result is equal to n minus 1, m minus 1. If you implement this as a recursive function, result r is equal to count n minus 1, j minus 1 plus count n, j minus 1. So 3 recursive calls here. It will be a nightmare. Your program is not going to run for more than 3. I think the complexity is somewhere 3 to the n, because you're calling this will be the recursive call tree of this one is going to be branching factor of 3. You're calling 3 recursive calls each time. Count this, and count that. And you have to also write the base case, but implementing this recursively, you will not going to be able to solve this problem for practical. If I give n is equal to 20, m is equal to 30, you will not be able to spit out any answer. So what we do is we write this as a dynamic program, which means that instead of having a recursive algorithm, we compute this from bottom up, not top down. For example, what is the number of paths that start at this cell and end at this cell? It's 1, right? There is only 1 path. If you start at the top left and end at the top left, there is only 1 path. And this 1 actually is the same for the first row. Let me just get to a new slide because it's really like it looks bad. So let's write it here on a clean sheet. So this one is 1. What I said is all the cells on the first row are all 1, right? Why is it 1? Because first row and first column, the only possible way, the previous, is it possible to come from, is there a cell here? No. Is there a cell here? No. So the only possible way to come is this. There's only one way to end up here in this cell. And that's why it's 1, 1, 1, 1. So all we do is we initialize this table like this, and then this will be much simpler, right? 3, 5, 7, another 5, 13, this one, 25, all I do is, it's really easy to fill in this table, right? I'm just, this 25, I'm just adding these three numbers. So you can just write a for loop. If you have a sequence of length n and length m, you can write a for loop, and your algorithm is going to run in polynomial amount of time, in O(n)m time and O(n)m space. If you're storing all these, you will figure out what this number here is very fast. Okay, computing it will be very fast. What about the quantity of this number? It's going to be huge. It's going to be a huge number. Okay, usually in questions like this, when you're asked a question when n is like 90 or m is like maybe, or I mean, one of them maybe 900, the other is 1000, the number you're going to get here is not going to fit in any integer limit. Usually when you're asked something like this as a question, you're asked what is the number in mode 1,000,001, or what is the last 7 digits of this number. We will be able to maintain the last 7 digits or so. So this is really a huge number. And the reason I made this introduction was to show you that you should actually know what O means. So this is the big O notation, the complexity, the time complexity of this. If you don't know the complexity of notation, you should either read some algorithms book to catch up, okay? Because I will be mentioning these a lot. So I will be talking about time complexities and space complexities. So algorithms is like a prerequisite to this course. So I will be talking about the time complexity. O(n)m means that in big O notation, the complexity is this polynomial, which means that this is the upper bound. The actual complexity is under this one. So when you multiply this nm with a constant, with a constant number asymptotically, so this is basically an asymptotic analysis. When n and m goes to infinity, this function, when multiplied by a constant, is always going to be on top of the actual running time. If the actual running time is fnm, this one, okay, this function, when I write O(n)m, like this O(n)m, this nm function actually, the function nm multiplied by a constant factor is always going to be on top of this actual function after some threshold. After some nm threshold, it's going to be above and it's going to remain above. All right, so this is like, then asymptotically, this means that its complexity is bounded by this. Okay, but if, as I said, if you have difficulties in understanding this, you either need to read some algorithm analysis book to catch up, or take this course after you take 301 data structures and algorithms service course if you're from another department. Okay, so yeah, the main reason I talked about this is very efficiently in O(n)m time. You will be in O(n)m space. So the size of the table is also O(n)m. When n is the number of sequences here, m is the number of sequences. The number of possible alignments is huge. So this solution that looking at all possible alignments and try to score them and pick the best one is not a feasible solution. But we can actually find a better one by using dynamic programming algorithm similar to the one that I just described here. So this was for computing the number of possible alignments. We are going to do something similar to this. We are going to use a setting that is similar to this one to solve the problem of what is the best way to, instead of accumulating all the paths, we can actually, with a similar setup, we will be able to maintain the best way to align these sequences. Okay, that's what the dynamic programming solution is. Now let's have a 10-minute break. Okay, I think it's a good time to have a break. Then I will go back to the slides and then describe the dynamic programming solution in the second hour and show you the code, the C code for that, and we'll see how it runs. All right, so I'm going to stop recording now, and we're going to meet at 10:45. 10:45 in 11 minutes. I will. Oh, okay. If you have questions, yeah, let me first stop the recording.